Hi, my name is Gianna Moulis. I am a biomedical sciences master's student. I am a member of Dr. I-Hsiu Huang's lab, and in our lab, we study the bacteria C. difficile. Previously, our lab collected environmental C. difficile isolates from various water sources in Taiwan, such as wastewater treatment plants. This is significant because it marked the first time that C. difficile had been isolated from a water-based source in Taiwan. Genotyping, ribotyping, and antibiotic susceptibility assays were performed for all isolates. My part in the study will consist of analyzing the phenotypic characteristics of these isolates, and the overall goal of this project is to generate a comprehensive profile for the C. difficile environmental isolates. This is a progress report for me, and today I will be reporting on colony morphology and gliding motility. In Table 1 here, I have listed the different ribotypes represented in my work. For some ribotypes, there is more than one isolate, but I chose to analyze only one isolate per ribotype. For colony morphology and gliding motility, R20291 and 630 were used as the control strains. Briefly, colony morphology was done by allowing a single colony of each isolate to grow on BHIA, and after the 7-day incubation period, the appearance of each colony was analyzed. Now, if you look at the bottom row, these colonies appear relatively smaller in size, while the colonies in the top row are larger, and they have these feather protrusions on the edges, which are indicating bacterial motility, which leads into the next part of this progress report. So next, I look at the gliding motility. And gliding motility, the purpose is to examine the ability of C. difficile to glide across an agar surface. These images were taken after a 5-day incubation period. For gliding motility, I measured the migration diameter at 24-hour intervals over this 5-day period. And if you notice here, ribotype 462 and AI83, these isolates are larger in size, and they have the feather protrusions on the edges indicating gliding motility. Same with the ribotype 060, while on the contrary, ribotype 106, these isolates are smaller in size, and there is little migration outside of the original inoculation zone. Next up, I wanted to determine if there are any significant differences between toxogenic and non-toxogenic isolates, and I did this by grouping the isolates into two groups. The toxogenic group had toxin genes, and the non-toxogenic group. No significant differences were seen in gliding motility between toxogenic and non-toxogenic isolates. To recap where I stand so far progress-wise, I helped establish a C. difficile isolate bank at LSU CHS. A wide diversity in colony morphology among the isolates tested was observed. The gliding motility on an agar surface was measured, and C. difficile displayed varying degrees of gliding motility. No significant differences between toxogenic and non-toxogenic strains were observed. For future work, we will next measure the swimming motility and sporulation efficiency. These are my references and acknowledgements, and I would like to thank everyone for tuning in and watching this video. Thank you all.